Okay, so welcome to this video. In this video, what we're going to discuss is drug addiction. Okay, right. So firstly, what I'm going to do is outline the structure then for this video. Okay, so firstly what we're going to do is we're going to define what drug addiction is. That's the first thing we're going to do. Then what we're going to do is take a step back. Okay, we're going to talk about the brain's natural reward system, okay, because it is this reward system that's going to be hijacked by all of the addictive drugs. Okay, so we're going to discuss the brain's natural reward system, and this is going to involve discussing quite a bit of neuroanatomy. Okay, so we're going to look at the important structures that are going to be involved in the brain's natural reward system. So we'll look at the ventral tegmental area firstly. Okay, we'll then look at the medial forebrain bundle, and then we'll move on to the nucleus accumbens. Okay, then what we'll do is we'll look at the theory of how this reward system works, and then we'll see how all of the drugs of addiction, okay, are going to hijack this natural reward system. And then what we'll do is we'll work through the main examples of addictive drugs. Okay, so we'll start with the psychostimulants, cocaine and amphetamines. Okay, we'll see how those interfere with this natural reward system. We'll go on to phencyclidine and ketamine. We'll talk about nicotine and many others as well, so opiates and cannabinoids and things like that. The one which we'll only mention, because we can't really say that much more about it, is at some point we will mention ethanol, or more commonly just known as alcohol. Okay, we can't really say much more about it other than how it hijacks the system. Okay, because not much more is known about alcohol. Alcohol's mechanism of action is very, very fuzzy. Even for a psychoactive drug, it's very, very fuzzy. Okay, right. So let's talk then about what drug addiction is firstly. So let's firstly define addiction. Okay, so addiction is defined as continuing to take the drug. Okay, so you continue to take the drug even though it's having negative consequences. So you continue to take drug even though you know that it's having extremely negative consequences on your life. Okay, despite the negative life consequences. Okay, so for instance, the financial consequences of taking the drug. Okay, and all of the side effects as well that the drugs often have. Okay, so despite the negative consequences. And we are now going to discuss what property that all of these drugs share which enables them to achieve this feat, that they drive you to continue taking them even though you know that they're having negative consequences. Okay, right. So we're now going to step back, as I outlined in our contents. Okay, we're going to step back and we're going to talk about the brain's natural reward system. Okay, and as I said in the outline of this video, we are going to have to discuss quite a bit of neuroanatomy in order to understand the brain's natural reward system. And the things that we need to know, okay, are what the ventral tegmental area is, which is often abbreviated as the VTA. We need to know what the medial forebrain bundle is, which is often abbreviated as the MFB, and we also need to know what the nucleus accumbens is, which is often abbreviated as NAC, like so. Okay, so we're going to go through each one of these structures. Okay, so we're going to start off then with the ventral tegmental area. Okay, the VTA. So I'm not going to assume that you know any neuroanatomy at all when we begin this video. Okay, so I'm going to talk you through from a picture that that I hope that everyone will be familiar with. Okay, so the picture that I'm going to start off with is the picture of what the brain looks like if you just take it out of the skull and look at it from the left-hand side. Okay, so let me draw this. So we are looking at someone's brain from the left-hand side. So here is the left cerebral hemisphere that we're seeing here. Okay, so we've got the temporal lobe sort of sticking out here, and then the rest of the cerebral hemisphere here. Okay, now pointing out from underneath the cerebral hemisphere, what you will then have is the lower portions of the brainstem. Okay, so here is a portion known as the pons. Okay, and then below the pons you've got a portion known as the medulla, and this is right in the midline. Okay, so it's coming out from the base of the brain, okay, right in the midline. Okay, and then it's continuous with the spinal cord below. Okay, so at this point it passes out of the skull through that large hole at the base of the skull known as foramen magnum. Okay, then also behind the brainstem, underneath the cerebral hemispheres, you have another portion of the brain known as the cerebellum, which literally means little brain. So let's label up the portions that we've got here. So this structure that I'm putting here, this is the cerebellum, which literally, as I say, means little brain. It's very, very important in the motor system, but we're not going to see it in this video because it's not thought to be involved in drug addiction. Okay, then we've got the spinal cord here. Okay, and this is the point where we enter the base of the skull here. There's a hole in the base of the skull known as foramen magnum. Okay, so I'll label that up as well. So foramen just means hole, magnum means big, so it's the big hole. Magnum. Okay, and then we've got the spinal cord, which will, of course, go down the vertebral canal. Okay, and we're only seeing the top of the spinal cord. It'll go on for much longer. Okay, right. Now, once you've passed through foramen magnum, the bit that's above the spinal cord, this is all known as the brainstem. Okay, the bit that's then entering the base of the cerebral hemispheres here. And there's more of the brainstem. We're only seeing the bottom of the brainstem. There's more of it that's embedded deep within the cerebral hemispheres. Okay, and we'll see more of this later on. Okay, so this is called the brainstem. Okay, now the first section of the brainstem, which I am colouring in red here, that portion is known as the medulla. Okay, so this portion in red here, this is the medulla. Okay, and above the medulla, you then have the pons, and we can only see the bottom of the pons here in blue. Okay, so this is the bottom of the pons here, and in a moment what we'll do is we'll take away the cerebral hemispheres, we'll take away the cerebellum as well, and we'll draw the rest of the brainstem, we'll draw the brainstem bigger, and we'll see the portions that are embedded deep inside the cerebral hemispheres. Okay, and then sitting on top here we have the cerebral hemispheres, and that's often called the cerebrum. Okay, so we've got the left cerebrum and the right cerebrum. We're looking from the left-hand side, so we're seeing the left cerebrum here. I'll just colour that in yellow. Okay, right. So, what we're now going to do then is we're going to imagine removing the cerebral hemispheres, and we're going to continue the brainstem on. We're going to see the bit of the brainstem that we can't at present see because it's deep within the cerebral hemispheres. Basically, it's buried deep within the cerebral hemispheres. So we're just going to continue the brainstem on, and we're going to go beyond the brainstem as well. We're going to see other surrounding structures that are above the brainstem and are also embedded deep within the cerebral hemispheres that we can't see at the moment. Okay, right, so let's now draw the brainstem out again then, without the cerebral hemispheres this time. So I'll start here. Let's say this is the spinal cord here. Okay, so this is the point where we go through foramen magnum, and the first portion of the brainstem that we then have is the medulla here. Okay, and then we've got the pons above that. Okay, and this is a little bit erect for the brainstem. I would have preferred it if it was a little bit more on a slant, but never mind. Okay, so here's the pons in blue here. It'll get the message across. Okay, so this is the pons. Okay, then we've got the medulla underneath here. Okay, and then the spinal cord beneath that. Okay, now we're seeing the top of the pons, which we couldn't see on this picture, and now above the pons what you're going to have is the next portion of the brainstem, which is known as the midbrain. Okay, so let's have the midbrain here in pink. So this is a new portion, so I'll highlight this up. And once again, you only have one midbrain, okay? So it's just like a cord at the moment. It's just like the continuation of the spinal cord. Okay, you've got one medulla, one pons, one midbrain. Okay, now the structures on top of the midbrain, suddenly that's the point where you go from having just one structure, okay, that's in the middle, okay, to having two structures, one on either side. Okay, so above the midbrain, what you have now perched on top here in an egg shape is the thalami. Okay, and notice I'm talking about thalami, plural. There is not just one thalamus. This is a common misconception, okay, in people who haven't studied neuroanatomy, okay, that there is only one thalamus. No, you have two thalami, okay, you have a left thalamus and a right thalamus. We are looking at the brainstem from the left-hand side, so this one that we are seeing will be the left thalamus, okay, and it's perched on top of the left side of the midbrain below. And on top of the right side of the midbrain, you'll have a right thalamus, which we can't see because it's hidden behind this left thalamus here. Okay, so in turquoise here, this is the left thalamus. Okay, the other structure that I want to show now, okay, and we will get to the ventral tegmental area. We're going to, in a moment, cut across the midbrain and see where the ventral tegmental area is, because it's going to be part of the midbrain. But I might as well use this opportunity to put a few more structures on that we're going to need later. Okay, so when we come on to the medial forebrain bundle, it's going to be very important that we have this anatomy, like the where the thalami are and where this structure that I'm about to show is. Okay, so sitting in front of the thalamus here, okay, like so, this is the hypothalamus, okay, with the pituitary gland coming down here and also the optic chiasm here. Okay, right, so let me show you the different portions then. So this isn't the hypothalamus here. That bit that I've just highlighted in yellow there, that is, I'll separate it off with the black pen as well. This is the optic chiasm. Okay, so this is where fibers in the two optic nerves cross over, okay, to go into the optic tract on the opposite side. Okay, in addition, this structure down here is not the hypothalamus either. That is the pituitary gland, and you've got the two portions of the pituitary, the anterior pituitary and the posterior pituitary. Okay, so this structure here is the pituitary gland, but the rest of that structure that I have just shown is the hypothalamus. Okay, so I'll colour in the hypothalamus in what colour have I got left? Orange we'll have. Okay, so here in orange, this is all the hypothalamus, and these little structures that you've got here, this little projection downwards, that's what's known as the mammillary body. That is part of the hypothalamus. Okay, you've got two of them in fact, one on either side, the left mammillary body and the right mammillary body. We are of course seeing the left mammillary body here. Okay, for such a tiny little structure there, it's surprisingly important. Okay, they're incredibly involved in memory of all things. Okay, so don't underestimate those little things there. Okay, now this is the basal surface of the brain. Okay, if you were to turn this structure that I've shown here, if you were to turn the brain over and look at the bottom, you would be looking at the bottom of the hypothalamus. You would be able to see the bottom of the hypothalamus basically, with the pituitary gland dangling down basically. Okay, if you'd manage to get the brain out very carefully. Generally, when people extract brains from human skulls, the pituitary gland ends up getting pulled off, basically. So usually you just have the pituitary stalk dangling there without the pituitary gland. Okay, but in principle, if you were to look underneath, you would see the bottom of the hypothalamus with the pituitary gland dangling down, and also the optic chiasm. That's very distinguishable. Okay, right. So now then, let's talk about the ventral tegmental area then. Okay, this is a part of the midbrain, this structure here. Okay, and for me to show you where the where the ventral tegmental area actually is in the midbrain, we need to look at the midbrain from a different angle. Okay, so rather than just looking at it from the left-hand side, as we have done here, what I now want to do is take a transverse section of the midbrain. Okay, and we'll take it up near the top. So we're going to take a transverse section like so. We're going to cut across like that, and we're going to look down from above and see what structures you have within the midbrain then. Okay, so the first thing is the midbrain has a rather more interesting shape than is visible when you just look from the left-hand side. Okay, so when you look from above, it looks like Mickey Mouse. Okay, and it really does look like Mickey Mouse. If you ever see it on an MRI scan, you'll be able to recognize it because you really do see Mickey Mouse looking back at you. However, when I draw it on paper, it never ends up looking like Mickey Mouse. Instead, it generally ends up looking like a pig. Okay, so the reason it looks like Mickey Mouse is it has the two cerebral peduncles here sticking out on either side, and those look like the ears of Mickey Mouse. Okay, so this is what the midbrain looks like from above. Okay, and once again I'll stress the point that you only have one midbrain. Okay, even though you can split it up into two halves and say this is the left side of the midbrain and this is the right side of the midbrain. Okay, and I should have just pointed out that this is the front here. Okay, this is the side that's towards your eyes. This is this side here. Okay, and this is the back here, towards the back of your head, and this side is this portion. Okay, so even though you can split it into the two halves, and people regularly do split it into the two halves, there is only one midbrain. You'd never hear someone talking about two midbrains. Okay, whereas you do hear people talking about two thalami. Okay, right. So let's talk about the landmark structures then of the midbrain. So firstly, you have these little structures that are at the back of the midbrain, which project outwards, okay, which I'm colouring in green. Now these aren't really visible on my picture here, but they should be. Okay, you should have little projections, and you have two of these, okay, at each level. So at the top level, which is the level that we're looking at, you have two pointing out from the back, a left one and a right one, and then underneath that you have two more basically, so overall you have four of the things. Okay, and the two that we're looking at, the ones that are higher up, these are known as the superior colliculi. Okay, and colliculi is the plural. Okay, the singular would be colliculus, so this would be the superior left colliculus, and this would be the right superior colliculus. And then the ones underneath, okay, that we can't see in this transverse section that I've taken, those are known as the inferior colliculi. Okay, so here are the superior colliculi here. Okay, and these are the ones that we're seeing. Right. Okay. Then another important structure, okay, is the periaqueductal grey matter here, which has a very important structure known as the cerebral aqueduct running through it. So this little tube right at the centre, this runs through the midbrain, okay, like so, and it is full of cerebrospinal fluid, and it is known as the cerebral aqueduct. Okay, so that's one of the landmarks of the midbrain, and we'll come back to this because when we see the third ventricle, which we will do later on, we will see that the cerebral aqueduct is going to be in continuum with the third ventricle, and it's actually going to be in continuum with the fourth ventricle, which is going to be below here. Okay, so it's going to go into the fourth ventricle, which is kind of here in position, okay, sitting behind the brainstem and in front of the cerebellum, okay, but it is full of cerebrospinal fluid, okay, and as I say, we will see the structure sitting above it that it's in continuum with in a moment. Okay, right. Then the grey matter that surrounds the cerebral aqueduct, which is part of the midbrain, is known as the periaqueductal grey. So peri means surrounding, okay, aqueductal means pertaining to the cerebral aqueduct, and then because it's grey matter it's called the periaqueductal grey, or for short the periaqueductal grey is often abbreviated to PAG. Okay, so we'll colour in the periaqueductal grey here in orange. Okay, right. Now, next up, what I'm going to show is the red nuclei. Okay, and I'm showing the structures that you need to show in order to make it look even vaguely like Mickey Mouse. Okay, so basically, here and here you have two structures known as the red nuclei. Okay, so this is the left red nucleus, and this is the right red nucleus. Okay, right, and I will colour in the red nuclei in red. Okay, now, you can hopefully see how it looks like a pig at the moment, okay, but if you actually do see it on an MRI scan, it really does look like Mickey Mouse. Okay, right, so, what we what were we actually after? Why were we doing this in the first place? Oh, we were after the ventral tegmental area. Okay, so this is going to ruin the picture. Okay, sitting here, basically, now it's got a third eye, okay, you have the ventral tegmental area, okay, so you only have one of these, okay, because it sits right in the midline. Okay, so this is the ventral tegmental area, and this is going to be so important for the brain's natural reward circuitry, and therefore it's going to be so important to an understanding of drug addiction. So VTA, what does that stand for? And this stands for ventral and then tegmental area. Okay, and for short, ventral tegmental area is often abbreviated to VTA. Right, so that's that little section of the midbrain there. Okay, so why is this area so important? Well, basically, this special area of the midbrain contains neurons which produce the neurotransmitter dopamine. Okay, now, I want to say something about dopamine, okay, because dopamine is not a normal neurotransmitter in the brain, okay, so, let me firstly show you the structure of dopamine, and then I'll tell you about the role of dopamine in the brain, and I will contrast the role of dopamine in the brain to the role of something like glutamate within the brain, or GABA. Okay, right, so dopamine then. So, firstly, the structure then of dopamine. So, dopamine, as the name suggests, has an amine group in it. Okay, so let's have a look at this. So basically, here is the amine group. We'll start by showing that firstly. Okay, and then off the amine group, you then have an ethylene group, which is just two methylene groups joined together. So there's one methylene group. Here's a second methylene group. Together they're known as an ethylene group. Okay, and then off the ethylene group, what you then have is your benzene ring. Okay, so here are the alternating double and single bonds, and then off the benzene ring you have two alcohol groups, one here and one here. Okay, so that then is the structure of dopamine. Okay, now let me highlight one important part of this structure. So an important part of this structure is the benzene ring with two alcohol groups coming off it. Okay, there is a special name for a benzene ring with two alcohol groups coming off it. It's known as a catechol ring. Okay, and it is for this reason that dopamine, along with many other neurotransmitters such as adrenaline and noradrenaline, collectively are known as the catecholamines, okay, because all of them have a catechol ring within them, along with an amine group. Okay, so dopamine, you would describe it as a catecholamine. Okay, the catechol, refers to the catechol group, okay, and the amine refers to the amine group that you also have. Okay, so that's the structure of dopamine. Now, its role as a neurotransmitter is very different from the role of glutamate and GABA. So let me just firstly remind you of the way glutamate and GABA work. Okay, and then we'll contrast the way that dopamine works in the brain. Okay, and the first thing actually to say is that you have hardly any neurons in the brain which actually use dopamine. Okay, so the ventral tegmental area is extremely rare in containing neurons that produce and use dopamine as a neurotransmitter. Most of the neurons of the brain use glutamate and GABA. So let me talk to you about the way that glutamate and GABA work. Okay, so basically the general principle is this. If we draw a little neuron here, okay, so neurons generally have a structure something like this, okay? So you have the cell body with lots of dendritic processes coming off, okay? So these little projections coming off, these are known as dendrites, okay? And here's the cell body with the nucleus in the center. And then you have your axon coming off the the cell body as well, and it ends with an axon terminal. Okay, so basically this neuron has these dendrites, okay, so these are called dendrites, and the dendrites are going to collect information from loads of other neurons, okay, so what happens is the dendrites have loads of little processes coming off them, okay, that look like this. And these little processes coming off the dendrites are known as dendritic spines. Okay, and I want to stress this picture is completely out of scale. The dendrites are humongous great things, and the dendritic spines are tiny little processes coming off them. Okay, so in reality what happens is neurons look more like this. This is the cell body, and then you've got the dendrites coming off, and they'll look more like this. Okay, and then you'll have the axon coming off like so. Okay, and then all over these dendrites you'll have tiny little processes which are going to be the dendritic spines, which will be forming synapses with the axon terminals from other neurons. Okay, so this picture is nice to draw, but it's not accurate. When you look at the histology, it looks something more like this. It's just this picture looks more difficult to understand. Okay, right. So basically what we've got here then is an axon terminal, okay, shown much smaller than the axon terminal of this one, evidently. Okay, so here in green is some other neuron coming along and synapsing onto the dendritic spine on the dendrite of this neuron here. Okay, so there's the dendritic spine in red. Now, this neuron will have a huge number of synaptic contacts like this, and there is a special name for these sorts of synapses. They're known as neural synapses. Okay, right. So the neuron will have a huge number of these neural synapses all over it, and if the synapse uses glutamate or GABA, okay, what will happen is these neurotransmitters are going to influence how likely this neuron is to fire an action potential itself. Okay, so basically we have these two major neurotransmitters within the brain. Glutamate is one, and then GABA is the other. Okay, gamma-aminobutyric acid is what GABA stands for. Okay, now glutamate is the main excitatory neurotransmitter of the brain. It makes neurons more likely to fire action potentials. So if this neuron wants to excite this other neuron to fire an action potential, it will release glutamate onto this dendritic spine. Okay, and glutamate will bind to glutamate receptors on the surface of the dendritic spine. Those glutamate receptors will open and they will allow in a current of sodium ions. Okay, so you'll be getting in sodium ions. Okay, now potentially we have another synaptic contact then. So we have another dendritic spine over here, and we have another axon terminal from a neuron here, and let's say this neuron wants to inhibit this neuron from firing an action potential, okay, when it's active. And I should stress that you can either be an inhibitory synapse or you can be an excitatory synapse. This neuron can't decide, oh, do I want to release glutamate or do I want to release GABA? It's one or the other, basically. So always a certain axon terminal will either inhibit or excite the postsynaptic neuron. Okay, so let's say over here we have an inhibitory neuron, okay, which wants to inhibit this neuron. It will release GABA onto the dendritic spine here, and that will allow in chloride anions, okay, which have a negative charge. Okay, so GABA will act on its GABA A receptors, okay, which will open and they'll make a chloride channel through the cell membrane, which will allow chloride in. All of this information will come together, so you'll be getting sodium and chloride anions from all over the different dendritic spines, all over the different dendrites. They'll all be coming together at this special area at the beginning of the axon here, okay, which I'm now highlighting in turquoise. Okay, so this special area at the beginning of the axon terminal here, this is known as the axon hillock, okay? And what will happen is the axon hillock will put all of the information regarding sodium ions and chloride anions together and decide whether it wants to fire an action potential, and then it will either fire an action potential or not. Okay, so that's glutamate and GABA. Okay, and those neurotransmitters are all about deciding whether the next neuron along is going to fire an action potential or not. Okay. Dopamine is completely different to this. Okay, dopamine has a much more broad regulatory function. Okay, the receptors that it's going to act on are not ligand-gated ion channels. Okay, so remember that glutamate receptors, okay, and the GABA receptors were ligand-gated ion channels, okay, which means that they were ion channels which were usually closed, but when the ligand became available, when glutamate or GABA became available, then they opened. Okay, now there's another name for ligand-gated ion channels that's often used. They are known as ionotropic receptors. Okay, because when the ligand is present, it's going to change the activity of the receptor, and the change in activity of the receptor is an ionic change, okay? It opens a pore through which ions can move. Okay, so you can either call ligand-gated ion channels or ionotropic receptors. Okay, the receptors for dopamine are not ligand-gated ion channels, not at all. They are G-protein coupled receptors instead. Okay, so let me talk about the receptors for dopamine, and then I'll talk about the nature of dopaminergic synapses. Okay, so the receptors for dopamine are what are known as G-protein coupled receptors, or GPCRs. Okay, so this stands for G-protein and then receptors. So G-protein coupled receptors. And there is another name for G-protein coupled receptors. They're also called metabotropic receptors, okay, to contrast them to ionotropic receptors. Basically, when dopamine acts on its receptors, it does not produce a change in the ion permeability of the cell membrane. Instead, it produces a metabolic cascade within the cells, hence why they are called metabotropic receptors. Okay, it causes a cascade, a signaling cascade of proteins, basically. Okay, right, so let me talk about what G-protein coupled receptors actually look like, or what metabotropic receptors actually look like. So basically there are a huge number of G-protein coupled receptors in humans. Okay, so in humans alone we have found over 800 G-protein coupled receptors. Okay, and many of them we still don't know what their ligands actually are. Okay, and they're known as orphan receptors. Okay, I think in the entire animal kingdom, okay, we've found now over 30,000 G-protein coupled receptors. So there are a lot of these things all over nature. Okay, and let me show you their structure. So basically, if I use these two parallel lines here to represent the inner and outer leaflet of a cell membrane. Okay, so remember, cell membranes consist of two layers of phospholipids. Okay, so this is the outer layer of phospholipids here, known as the outer leaflet, and this is the inner layer of phospholipids here, known as the inner leaflet. Okay, so G-protein coupled receptors then, they are integral membrane proteins. And basically, if this is the amino terminus here, the characteristic feature that they all have is that the polypeptide spans the membrane 7 times. Okay, like so. So they are a single protein, which is actually something that separates them from the ligand-gated ion channels. The ligand-gated ion channels are actually made up of multiple proteins all stuck together to make the full ion channel. Okay, G-protein coupled receptors are a single polypeptide. Okay, so they have an amino terminus and a carboxylic acid terminus. So they have these 7 membrane-spanning alpha helices. That's the characteristic feature of G-protein coupled receptors. And when their ligand binds to them, it activates signaling cascades within the cell. Okay, right. So there are 5 different types of dopamine receptor. Okay, and these are called the D1 receptor, the D2 receptor, the D3 receptor, the D4 receptor, and the D5 receptor. Okay, so they are all G-protein coupled receptors, so they all have the same basic structure where they have 7 membrane-spanning alpha helices. Okay, but they will of course all be different proteins, so they'll all have different sequences of amino acids. Okay, it's just they all have this common membrane-spanning topology. Now they're grouped into 2 separate groups. Okay, D2, D3, and D4 are collectively known as the D2 type dopamine receptors. Okay, and D1 and D5 are collectively known as the D1 type dopamine receptors. So let me highlight those up now. So D1 and D5 together in blue here are known as the D1 type dopamine receptors. Okay, and the way that these two different clusters of dopamine receptors differ is in the downstream pathways that they actually activate. Okay, so D1 type dopamine receptors activate the GS heterotrimeric G proteins and hence stimulate adenylyl cyclases. Okay, the D2 type dopamine receptors activate the GI heterotrimeric G proteins and hence they inhibit adenylyl cyclases. Okay, so very different pathways that you're activating. Okay, right, so dopamine receptors are completely different from the receptors for glutamate and GABA that are being used at the sort of classical idea of a neurotransmitter synapse. Okay, right. The other thing that I've said is that you have very few dopaminergic neurons in the brain, okay, and they are not just scattered around homogeneously, okay. Instead, you have specific areas in the brain which have all of the dopamine neurons clustered in them, okay, and one of these special areas is the ventral tegmental area that we've seen, okay, and what's going to happen is it's going to send its axons all over the brain, okay, so it's got loads of neurons in it, and different neurons in the ventral tegmental area are going to send their axons to different places so that overall the ventral tegmental area has axons flowing out of it all over the brain, delivering dopamine to areas all over the brain. Okay, now what I want to talk about also is the different types of synapses that these axon terminals of dopaminergic neurons can make with cells. Okay, so they can make neural synapses, just like this sort of classical setup we've seen here. Okay, so let me show this. So what you can have is these dopaminergic axon terminals synapsing on dendritic spines just as though they were a normal axon terminal releasing either glutamate or GABA. Okay, but this is a very special axon terminal. It releases dopamine. Okay, so it will release dopamine onto the dendritic spine here, and by the way, the abbreviation for dopamine is DA, so it will release dopamine onto the onto the dendritic spine, and now instead of the dopamine acting on the ligand-gated ion channel, it will activate one of these dopamine receptors that we've discussed, and that will trigger a signaling cascade inside the cell. Okay, so different from the changes in ion permeability that are contributing to whether the neuron will fire an action potential. Okay, so this signaling cascade won't change whether it's going to fire an action potential or not. It will have broad consequences for the neuron's function, however. Okay, so it will have generally longer lasting consequences and more generalized than just a little contribution to whether or not the neuron's going to fire an action potential. Okay, right, so that's one of the types of synapses. When you have an axon terminal synapsing with a dendritic spine like that, that's known as a neural synapse. Okay, so this is a neural synapse, this is a neural synapse. Contrast that to other types of synapses that dopaminergic neurons can form. Okay, so the other type of synapse I'm going to talk about, in fact I'll show it here, is what's known as an en passant synapse. Okay, now what does this mean? Well, this means in passing synapse, okay, that's what this means, in passing. Okay, now basically this doesn't involve a an axon terminal synapsing onto a dendritic spine. Instead, generally what happens is, in fact I will show this down here, generally what happens is you'll have a dendritic spine, like so. So this is a dendritic spine magnified up largely, okay, and this will have some axon terminal synapsing onto it in a neural synapse, and generally this axon terminal will be releasing something like glutamate or GABA. Okay, so this is either glutamate or it's GABA. Okay, one or the other, not both of them. Okay, now then what you'll have is one of these en passant synapses, okay, on the sort of neck of the dendritic spine. Now what is an en passant synapse? Well, basically the idea is that instead of you having the axon terminal synapsing onto the dendritic spine, instead what you have is these special structures known as boutons, okay, along the axon length. Okay, so if this is our neuron, let's say this is our dopaminergic neuron, so I'll draw it out here, and this picture will be hugely exaggerated. Okay, so here is the cell body of the dendritic neuron, and the idea is that then along the axon you have these special sort of swellings known as boutons. Okay, and you'll have loads of these, so this is known as a bouton. Okay, and basically the action potential will propagate along here until it gets to the bouton, and then from the bouton you will release dopamine out, and then the action potential will go on to the next bouton, cause dopamine to be released from that bouton, and so on. Okay, so rather than just having an axon terminal at the end which will release the neurotransmitter, you have loads of these in-passing synapses. Okay, so this is supposed to be a bouton of a dopaminergic neuron here, releasing dopamine onto the neck of a dendritic spine, and here then you'll have some of these dopamine receptors which will be modulating the function of the dendritic spine, okay, even though they don't directly change the ionic permeability of the cell membrane. Okay, right, so this is one of these boutons here in red which will be releasing dopamine. So you have a lot of dopaminergic neurons forming these en-passant synapses with other neurons. Okay, and it means that they can form connections with a huge number of different neurons and release dopamine onto a huge number of different neurons, modulating the function of those neurons. Okay, in addition, going even further, we've now seen neural synapses en-passant synapses. The other thing that dopaminergic neurons can form is what's known as varicosities. Okay, now this is when the dopaminergic neurons don't actually synapse onto anything really. Instead, what you have is just an axon terminal, like so, of the dopaminergic neuron without any postsynaptic structure. And instead, what happens is the dopaminergic neuron, when it fires an action potential, so if an action potential comes along, you just release dopamine into the extracellular fluid now, and that dopamine can just broadly act on any neuron that has a receptor to it that's in the vicinity, basically. Okay, now when you have an axon terminal that just ends without having a clearly defined postsynaptic neuron, that's known as a varicosity, so this is a varicosity here. Okay, right. So, basically, what was the point of all of that? My point was that the ventral tegmental area is sending out axons all over the brain, okay? And it's releasing dopamine all over the brain. Now, sometimes it's releasing dopamine onto actual neurons, actual specific neurons, either in the form of these neural synapses, where the axon terminal synapses specifically onto a dendritic spine, or in these en-passant synapses, where boutons synapse generally onto the necks of the dendritic spines, regulating the function, certainly of that dendritic spine, but of the neuron more broadly going onwards, okay, so not just the dendritic spine, because we've got the dendrite here, okay, so it'll be modulating this local function here. Okay, and we've also got these varicosities just releasing dopamine into the extracellular fluid of the brain, and that can act on any neurons in the vicinity. Okay, so really, you're just releasing dopamine potentially all over the brain. And what this system is all about, what the dopamine system as it's called is all about, is modulating whole brain functions rather than modulating the behavior of one neuron, which is what all of this is about. Okay, and it's ghastly important, but it really is just the modulation of one neuron, and you have to build 100 trillion of these things together to actually get something that's incredible. Okay, the dopamine system really is modulating the function of the entire brain, okay, because it is releasing dopamine onto the entire brain, okay, and therefore it's controlling the amount of dopaminergic input that the entire brain is getting, okay, and this controls broad regulatory functions, basically. So for instance, it controls things such as arousal along with other monoamine systems. So there are other systems in the brain that are just like this, where you have specific little areas containing neurons which make the neurotransmitter. So important examples are 5-hydroxytryptamine, also known as serotonin, and also noradrenaline, NA. Okay, these are what are known as the monoamines, along with dopamine. Dopamine is also a monoamine. It just means that you have a single amino group in you. Okay, so these are the monoaminergic neurotransmitters. Okay, and basically what happens is you have special areas in the brain which produce these neurotransmitters, and then other areas of the brain don't use them whatsoever. Okay, so what happens is these specialized areas of the brain send their axons all over the brain and release these monoamines onto all the different areas of the brain, and therefore these single areas control how much of the monoamine the entire brain gets. Okay, and through this they can modulate entire brain functions, such as your level of arousal, i.e. whether you are extremely vigilant, okay, or whether you're about to fall to sleep. Okay, right, so those sort of things are controlled by the monoamine systems, of which the dopamine system is one. Okay, right, and we're going to see another huge function of the dopamine system, which is its control of the reward system within the brain. Okay, right, so we have now talked about the dopamine system. What we're going to now do, well, we haven't talked about the dopamine system, we've talked about it broadly. Okay, what we're now going to see is the specific places where dopamine axons are going to be sent from the ventral tegmental area. Okay, and the two places they're going to go are the cortex and also the nucleus accumbens. Okay, so let me get another piece of paper here. So there are two important pathways that we are going to discuss. We're going to discuss something called the mesocortical pathway, okay, which is the pathway by which you send dopaminergic neurons from the axons of dopaminergic neurons from the ventral tegmental area, which is in the midbrain, to the cortex. And the reason it's called the mesocortical pathway is that meso stands for mesencephalon, okay, which is another name for the midbrain. It's the fancier name for the midbrain. Okay, so remember the ventral tegmental area is within the midbrain, so these dopaminergic axons are going from the midbrain to the cortex, hence the mesocortical. Okay, so this is going to be the mesocortical pathway. Okay, so this is going to send dopaminergic axons from the ventral tegmental area to the cortex, and we also want to see a pathway known as the mesolimbic pathway, which is a pathway by which dopaminergic neurons send their axons to a structure known as the nucleus accumbens. Okay, so one of the key areas of what is known as the limbic system, which are areas involved in emotion, okay, or at least they were initially hypothesized to be involved in emotion, okay, is the nucleus accumbens. Okay, so this is a pathway from the mesencephalon to the nucleus accumbens. So we want to start off with the mesocortical pathway, and the reason we're going to start off with the mesocortical pathway is because it will allow us to develop the anatomy of the medial forebrain bundle on its own, and we can see it going to the cortex, okay? And we don't have to worry about developing the anatomy of the nucleus accumbens. So basically, the reason I'm picking the mesocortical pathway is because you already know what the cortex is, hopefully, okay? So I can tell you about that, and we can just focus on the medial forebrain bundle and where that is. And then we can go on to the mesolimbic pathway, we can discuss where the nucleus accumbens is, and then we'll already know where the medial forebrain bundle is. So it just means that I'm not having to do two things at once. We can do it nice and in order. Okay, right, but we'll do that in the next video.